guys, and welcome to IB Online. Javed Khan Afridi here, and you are watching A is Level Biology. In the previous class, we discussed about the interface and the process of mitosis, but not in detail. I just told you that interface is one of the longest phase of cell cycle, and it occupies about 80% of the total time. If one of the cell takes 20 hours to complete one cell cycle, then 16 hours will be spent in interface. And the remaining 4 hours occupied both the mitosis as well as cytokinesis. We discuss about the interface, that there are three steps in the interface: the G1, the S phase, and the G2 phase. I told you that G1 is one of the longest phase of the interface. And in G1, what's going on? We discuss all the aspects of the G1. Then I told you about the S phase, where the replication of DNA take place. And then the G2, of course, where some expression of genes and some of the important protein has been synthesized, and these proteins are later on used in the process of mitosis. I told you that once the interface is completed, then the process of mitosis is going to start. And I told you that mitosis further consists of four phases, which is prophase, metaphase, anaphase, and telophase. I told you that prophase of mitosis is the longest phase, while anaphase is the shortest phase of mitosis. The entire process of mitosis takes 17% of the 20%, and 33% is the process of cytokinesis, which consume the time. And the interface, three phases, cell cycle, the G1 phase, S phase, and G2 phase collectively occupied 80% of the time. It means that the cells most of the time remains in the form of interface. And that's why whenever you see some of the meristematic region or some of the stem cells which are in the process of division, you can see that most of the cells will be in the steps of interface, because it takes longer time, and the other steps remains for a short period of time. So mitosis and cytokinesis, collectively known as cell division, because in mitosis, nucleus is divided. But in cytokinesis, cytoplasm is divided. When you are dividing the cytoplasm, you are making two cells from one cells. And before the division of cytoplasm, we divide the nucleus into two. So in one cell, we have two nuclei. Once there is one cell, but we have two nuclei, it means that's the end of mitosis. And then immediately we split the cell in the middle, separating two units of the cell. Each one has the same number of organelles, same number of cytoplasm, same contents of cytoplasm, and same DNA contents, which is cytokinesis. So cell division is something different from the cell cycle. Cell division comprises mitosis and cytokinesis, while cell cycle comprises all the three steps, including interphase, mitosis, and cytokinesis. In other words, we can say that mitosis and cytokinesis these are the steps of division of cytoplasm and division of DNA, while interphase is not the division, it's the duplication, it's the doubling of the content. So first the content should be doubled, either they are the cytoplasm or they are the DNA. And then later on, they will be split down so that the original number of all the components will be restored in the daughter cell. We have discussed all things. Now today, I'll tell you what are checkpoints. Checkpoints are certain types of series of protein molecules which identify each step of the cell cycle. For example, if the cell cycle goes to complete the interphase, then there are some protein molecule which check that either all the content have successfully doubled or not. If the DNA or cytoplasm or protein synthesis not fully developed, then it will arrest the cell and prevent it to move into mitosis. So it will prevent the further step. So checkpoint basically some protein, signaling protein, which identify either all the things have been taken, all the things have been going in the correct way, then they allow the cell to move in the next step. Otherwise, it will prevent the initiation of the next step. So I'll going to discuss with some more detail about the checkpoints. So let's stay with us, and I'm going to explain it some more. So checkpoints, I told you, are signaling protein molecules, and you know that we have three types of checkpoints. There are three checkpoints in the entire process of cell cycle. So one is called G1 checkpoint. One is called G2 checkpoint, and one is called M checkpoint. Where is G1 checkpoint? At the end of G1 phase of interphase. So when interphase is in operation, during interphase, at the end of G1 phase, when G1 is going to finish, the G1 checkpoint will be in working. It will start the working, and it will observe all the aspects. Now you must know what was going on in the checkpoint. If you remember, I have mentioned some important point: doubling of the organelles, doubling of the cytoplasmic contents, synthesis of the nucleotide, expression of certain genes, translation, and to make the required protein necessary for the process of cell growth. All these substances should be checked, either they are taken place correctly or not. The signaling molecule will proceed, and they will see either if all the organelles are doubled correctly, all the cytoplasm contents have become doubled or not. We have adequate number of nucleotides for the next phase. I mean, when the replication of DNA will be proceeding. Either all the genes have been expressed to form the required proteins. If all these are okay, I mean, yes, the process have been completed successfully, then they will provide a green signal, and they will say, okay, everything has been taken place correctly, and now proceed the cell into the next step, which is G2. So at the end of G1 phase, there is the checkpoint, G1 checkpoint. And I told you checkpoint will identify all these things. If they are okay, then the cell will proceed. But if they are not, I mean, the DNA is not completely doubled, cytoplasmic contents are in irregularly pattern, organelle have been not doubled anomalously, so it will arrest the process, and the division will be stopped. They will not move to the next step, which is the S phase. Instead, the cell will be arrested, and it will move back into another step, which is called G0 phase. Now, what is G0 phase? G0 phase is just like a workshop of the automobile. If there's some error, some mechanical error in a car, in a vehicle, so you just have to move in a workshop for fixing. So this is a fixing area where the cell, which is not in proper doubling, so it may be because of some error. So this error is going to be fixed in G0 phase. If it is fixed, then it will move to again S phase directly, because it will be superpassed the G1 phase and it will be corrected from G0 to S phase. But if it takes longer time, it will not fixed in the workshop, not repairable, not correctable, then it will be damaged, then will be destroyed by the body immune system, and the cell will not be completed. So there are some of the cells which are not in normal rhythm, so they will be destroyed by the body immune system. So G0 phase is one of the phase where some of the repairable cells remains for a short period of time. If they have been corrected, then they will move to the next phase. So we can say that G1 phase is a reversible phase. I mean, if one of the cell have been completed the G1 phase, but the checkpoint identifies some error, so they will be moved back to G0, mean they will be reversed, not necessarily that the cell will be proceeding to S phase. Now, what about the G2 phase now? G2 phase is present at the end of G2. There is no checkpoint in between the S phase and G2 phase. So G2 phase, G2 checkpoint also contain series of proteins which check any errors in the dividing cell just at the end of G2 phase. Now, so many things will be checked at the end of G2. First of all, either the mitochondria have doubled or not, the centrosomes have been divided into two or not, the DNA have been successfully semi-conservatively replicated or not, do we have a double contents of DNA or not, do we have adequate number of the enzymes, the gene expression have been taken place completely, do we have enzymes for the digestion of metabolites to produce energy, to give us energy for the cells, for the process? Do we have the tubulin protein for the spindle fibers which are produced in the G2 phase? All these still will be identified in G2 checkpoint. If yes, then the cell will be proceeding and it will enter into mitosis, because G2 is the end of interphase. So the G2 signal means the cell has been successfully completed doubling of all the contents in the cell. And then it will move into the mitosis and will start the prophase. Now, it will move to prophase. At the end of prophase, then we start the metaphase. When metaphase has been completed, then there'll be another checkpoint which is called M checkpoint, M for metaphase or for mitosis. So M checkpoint is also series of protein complexes that become active at the end of metaphase, to check any errors in the previous steps of mitosis. The previous steps, what are the previous steps? It was prophase and metaphase. This is the last checkpoint. It means that at the end of anaphase and telophase, we don't have any other checkpoint. So if somebody asks you, which step of mitosis is irreversible? That's why we say that anaphase and telophase are the irreversible phases of mitosis, because there's no checkpoint at the end of anaphase and telophase, because checkpoint can check the errors. If there's any error, it can reverse the process. Like you just are moving from one country to the other country. So the airport border is the checkpoint where your security has been checked. So there's a chance if you have some types of illegal documentation or something which is prohibited in that country, you will be departed back from the airport. It means you have been reversed because there's a checkpoint. But you will cross all the checkpoints. Maybe you have been screened successfully. It means you just have to enter the country. The same way, the last checkpoint is at the end of metaphase. So we say that all the steps of the interphase and the first two steps of mitosis, prophase, metaphase, are the reversible steps. They can be reversed at any time if there's some error. But once the metaphase has been crossed, and there's all the checkpoints say, okay, then there is no chance of the reversibility. So we say that the first irreversible phase of cell cycle is the anaphase. Of course, the other one as well. This is what I want, what we have discussed about the checkpoints. So let's to explain some in more detail here. So what are the checkpoints actually, and what they are doing actually? So checkpoint, what are the checkpoints here? Checkpoint is one of the several points in the eukaryotic cell cycle at which the progression of a cell to the next stage in the cycle can be halted. If there is some error, it will be halted. Halted means prevented, arrested, until conditions are favorable. Conditions are favorable means sometime there's no error, everything is going okay, but we don't have the adequate number of nucleotide, we don't have sufficient diet to do the process of cell cycle. So some of the things, some of the materials, some of the chemicals required from our diet in a particular cycle, if there's no appropriate diet, the cell cycle will be halted. I mean, the conditions are not favorable. So these conditions are checked by the checkpoint, which is a series of protein. This transition is controlled by some cell signaling molecules that regulate the cell cycle. So every checkpoint has some series of protein molecules which are acting as a signaling molecule to identify all these steps are okay or not. Now, the first one is the G1 checkpoint. The G1 checkpoint determines whether all conditions are favorable for the cell division to proceed. The cell will only pass the checkpoint if it is in appropriate size and it has adequate energy reserves. Energy will come from the ATP, and ATP is produced when there is some chemical reactions, and for the chemical reaction we need enzymes, and I told you that enzymes formation takes place in the first step, that's in G1, expression of genes. So there's no adequate enzymes, how you can get the energy, the process will be halted. At this point, the cell also has checked for the DNA damage as well. If a cell meets the requirement for the G1 checkpoint, the cell will enter S phase, means the green signal will be on, and begins DNA replication. A cell that does not meet all requirements will progress to the S phase, will not progress to the S phase, will be halted, it will be arrested. The cell can halt the cycle and attempt to restore the problematic condition, I mean the repairing of that problematic conditions, or the cell can still advance into G0 in inactive phase. The cell become inactive, it will stay in G0 stage until the conditions are not favorable or the problem is not fixed, and await further signal when condition improve. So this is G1 checkpoint and it locates at the end of G1 phase. G2. The G2 checkpoint ensure all of the chromosome have been replicated, because replication of DNA takes place in the S phase and it is located at the end of G2. So it also checks the S phase components and that the replicated DNA is not damaged before cell enters mitosis. If the checkpoint mechanism detect problem with the DNA, the cell cycle is halted and the cell attempts to either complete the DNA replication or repair the damaged DNA. If the DNA has been correctly replicated, now there is one of the molecules, signaling molecule, its name is cyclin dependent kinase, CDKs. CDKs are a kind of proteins, signaling proteins, signal the beginning of mitotic cell cycle. So it will trigger the process of mitosis to begin. And then finally the M checkpoint, the last one. The M checkpoint occurs near the end of the metaphase stage of mitosis. Now, until I'm not to explain the events of mitosis, you cannot understand what's going on in the M checkpoint, because M checkpoint is located at the end of metaphase. So it checks the things in both the metaphase and prophase to occur. So if you know what's going on in the prophase and metaphase, then you can identify the exact function of M checkpoint. However, I'm just going to tell you briefly here that the M checkpoint is also known as the spindle checkpoint, because it determines whether the entire sister chromatids are correctly attached to the spindle microtubules before the cell enters. So definitely you will not understand now because you have not studied the the metaphase, the spindle formation. I'll tell you in the next video. But here, this is the 3 checkpoints which are working together. Now, that's the end of the checkpoints. If all these have produced the green signals, the cells will enter in the process of progression in the next step. Now, it's the time to discuss about the mitosis. What's going on in mitosis? Because the interface has been completed, the first 2 checkpoints provide green signals, and now we have entered in the mitosis. I'm not talking about the M1 now, and I'm not talking about the M checkpoint now. I'm just talking about the G1 and G2. So at the end of G2 checkpoint, we have ensured that the interface has been completed successfully, no damage. And after G2 checkpoint, we entered in mitosis. Now let's study what's going on in mitosis. So once you identify the exact mechanism of mitosis, then definitely you will understand the concepts of M checkpoint as well. So let's see to discuss what's going on in the in the mitosis. So what's going on in mitosis? Yes, in mitosis, I told you there are 4 steps. And the first step we are discussing is the prophase. What's going on in the prophase? Prophase is the longest phase, as it's the major steps in the cell cycle. And what's going on in the prophase? Some important, prophase is a continuous process, but there are some important key points in the prophase which we have to highlight. So what's going on in the prophase? Let me to explain now. Initially, the chromosomes or the DNA and histone, which is collectively known as chromatin or chromosome, but it's in the uncondensed form, chromatin becomes condensed. The DNA goes to condensed. If this is the chromatin, like in this form, this is one of the chromatin, it's too thin and uncondensed structure. So it will be condensed like taken in this form. Now look at the difference between these 2 structures. It has been condensed one, and this is the uncondensed one. This is the uncondensed, and this is the condensed one. So once you compress a longer strand to shorten, so its length will go to its thickness. It becomes thick. Compressing, so it becomes thickened, means it becomes condensed. This is the condensed form. But as I told you that before mitosis, the DNA has been doubled. So this is one of the chromatin, the original one, and the other chromosome, I'm just making like in green, in blue. This is the another one which has been duplicated during the process of interface, and this also become condensed like in this way. See, these 2 have been condensed. Now 2 DNA molecules which are condensed. In our cell we have 46 chromatin. So all these 46 chromatins will be converted to 92 chromatins because it's been doubled at the time of interface. Because in interface, semi-conservative replication makes the DNA to be doubled. 1 molecule of DNA is converted into 2 DNA. So initially we have 46 DNA, and now at the end of interface we have 92 DNA. So here, initially we have the original DNA which I have shown in the red, the condensed one. We have 46 of these original DNA. But later on, we also have produced other DNA molecule by condensation, by the semi-conservative replication, the condensed one, and they also have 46. However, both of these 2 DNA at the time of prophase will join together at the point called centromere. Now, these molecules will become like in this way, just going to form, like they will unite together in this way. This is one of the original DNA, and this is the other DNA forms. They will join together to make a chromosome. This is called chromosome. Why is chromosome? Because they have one of the point here, the point of intersection just make a black one. This point of intersection where they are joined together. It's like the bigger one. I'm just going to draw it as this is one of the condensed DNA, the original one. And this is the other one, the condensed one. I will form two DNA been formed. Now that area where these two are connected with one another, here, the region where they are joined with one another. This is called centromere. Centromere. This centromere is also present on a single chromosome. Like if I'm telling you that one of the condensed chromosome is here, so it already have the centromere. If the centromere is present, then it is called chromosome. It not will be a chromatin. Keep in mind that one chromosome is represented by one centromere. I mean, chromosome is represented by condensed mass of DNA with histone showing a centromere. Centromere is a non-coding region on the DNA usually. I'll tell you more in detail in the next video. So here we have the one chromosome. But when they unite with the other one, like in this way, it also have its centromere, but the centromere now will be merged into one centromere. So if there's only one centromere, still we say this is also one chromosome. They are also known as one chromosome because they have only one centromere. But in this case, now each chromosome is called chromatid. Once they join together, they are known as the chromatids. In the separate form, they are known as the chromosome. Like in this form, each one is called a chromosome. Oh, this is a chromosome actually. This is the chromosome. These are the chromatin, uncondensed. Two chromatins here. Here's one chromosome because it's one centromere. And this is also called one chromosome. This is also one chromosome because they also have one centromere. The centromere join and merge together to make only one. So it means we say that chromosomes, some of the chromosomes have two DNA and some of the chromosomes have one DNA. This is the chromosome. This is the chromosome with one DNA. This is the chromosome with two DNA. But the chromosome which have two DNA, then one of the DNA molecule is called chromatid. In this case, they are called the chromatids. But when they are removing from one another, then each chromatid is said to be a chromosome. So whenever you see the chromosome in this form, they are attached together like in this format. So then it is said to be a chromosome and each partner is called sister chromatid. But when they get separated from one another, they are no longer remain a chromatid. They become the chromosome because each one of them have a separate centromere. Don't forget this point. If a DNA molecule is condensed and it has its own centromere, then it is a chromosome. It's not a chromatid. But if we have two DNA, both are condensed and they are unite together at a particular point, still it is one chromosome. But each of the DNA in this case is called a chromatid. So here in mitosis, which is the longest phase, the first thing that the chromosomes, the DNA become condensed. The DNA become condensed. The process of condensation begins earlier in prophase and still remain continue. You can't say that the condensation become complete. The condensation still remain continue in the entire process of prophase. So when you are in prophase, you can see the DNA in the condensed form. So we say that condensed DNA should be condensed. Each condensed DNA in the form of chromosome, because condensed DNA is called chromosome. Each chromosome have two DNA molecules it means the chromosome is like in this form, because it has two DNA molecule. In one chromosome we have one DNA. So in prophase you can see the chromosome like in the form of a double helical structure, although it's not be so much prominent, but it will be like somewhat double helical structure. Each chromosome have two DNA molecule. It means that each chromosome have two identical sister chromatids joined together by a centromere. Since there's only one centromere, so we say it's only one chromosome. It means like initially, if the chromosome like in this form, we have 46 of them. Similarly, the newly formed will be also in 46 form. It means overall these are 92. But in prophase, as I told you that they unite together at the metaphase. Now we have 46 of them, because 46 like this one and 46 this one. So now you have 46 chromosomes here. But before their union, we say that we have 92 chromosomes. They are not chromatid now because they are not unite together. When they unite together, then we say there's a chromosome. So here we have 92 DNA. Here we also have 92 DNA. However, here we say that we have 92 chromosomes, and here we say 46 chromosomes. But the chromosome number have been reduced to half. However, each chromosome have two DNA. So actually the number of chromosome, in the sense of chromosome, we say 46, but originally it had the same DNA as in 92 chromosomes. So 92 chromosomes and 46 chromosomes both have the same DNA. The difference is only that in case of 92, each chromosomes contain one DNA, but in 46 chromosome, we have two DNA molecule. That's why we say that the semi-conservative replication keeps the DNA contents double, and the DNA after doubling become condensed, and after condensation they join at the point of centromere to make sister chromatids. And all these things are taking place in the prophase. So here we say that joined together by a centromere. There is one important area, that the nuclear membrane is going to be disintegrated. Nuclear membrane is disintegrated, or you can say is fragmented. Nuclear membrane is fragmented and then disappeared. I mean, if there is a nuclear membrane and inside we have the condensed DNA, here we have the condensed DNA. So this is the original number of DNA, and then after replication, new DNA is also joined with here in the condensed form. So we have total of 8 DNA molecule and 4 chromosomes here. So here we have 4 chromosomes, 8 chromatids, 4 centromeres, and 8 DNA molecule. Or you can say 16 DNA strands. because one DNA molecule have two DNA strands, double helical structure. So if there are 8 DNA, we have 16 polynucleotide or 16 DNA strands. We have 4 centromere. You can see only 1 centromere where they are fused together. So each chromosome has 1 centromere. We have 8 chromatids because one chromosome is made up of 2 chromatids, and we have 4 chromosomes here that contains 1. Now in this case, what's going on, that initially the nuclear membrane is disintegrated. It's disintegrated like in this way. It is fragmented initially, and this fragmentation increases gradually more, then it finally disappears. The nuclear membrane is finally disappear, and all this going on in the prophase. Then one another point here, disappearance of nucleolus. Nuclei are also disappear from the nucleus, and you say that nucleolus is one of the spherical structure inside the nucleus, it will be disappeared. So nucleolus will also disappear. Nuclear membrane disintegrated, nucleolus disappeared, chromosome become condensed. Each chromosome is made up of 2 DNA molecule joined by centromere. Then the 2 centromeres, also sorry, not centromere, the 2 centrosomes. The 2 centrosomes, the organelle I told you that has become double in interphase. Centrosomes, some kind of the organelle located just near the nucleus, having a condensed mass of cytoplasm, and inside the centrosome we have centriole. I told you the important role of centriole to develop the spindle fiber, and this spindle fiber pull apart the chromatids to split the chromosome. So the 2 centromere, the 2 centrosomes are migrated to the opposite pole of the nucleus. Opposite pole of the nucleus. What does it mean? Opposite pole of the nucleus. I mean, here we have the DNA. As the nuclear membrane have been disappeared, initially we have the nuclear membrane before the disappearance. We also have the centrosome here in the nucleus initially. The nucleolus inside. And we have the centrosomes just near the nucleus centrosomes, and we have 2 centrosomes actually, because the centrosome also divided in the interphase from 1 to 2, and in the centrosomes we have the centrioles here. Now in prophase, what's going on, that initially the nuclear membrane is going to disintegrate, as I told you. When the DNA doubling occurs, then the nuclear membrane slightly is going to be disintegrated, and then finally it completely disappear, as I told you. Then the nucleolus is also going to be disappear, and now these 2 centrosome, which are already present in pair, they will be migrated to the opposite pole. So they will move like in this way. Now this is the pole area. As like in geography, that the earth like showing here like in this film format. So it says this is the equator. This is the equator, and these are known as the poles. So here we use the word poles for the nucleus as well. This is one of the pole, and this is the other pole. So the 2 centrioles are migrated to the opposite poles. Then finally at the end, when we are near to the end of the prophase, just quite at the end of the prophase and then the metaphase beginning, what's going on, that spindle formation are going to be formed. Spindle formations are protein fibers. Protein fibers are made up of from a simple protein called tubulin. So tubulin protein, like this is one of the tubulin molecule. I told you in the first chapter that tubulin is a kind of globular protein, and in two forms, and there are two forms: alpha tubulin and beta tubulin. And this tubulin, like in the form of globular one, this is one of the tubulin, and the other one is just going to be other color, and they're joined together. Now, these two tubulin are called collectively, they are known as the dimer. I told you initially in the first chapter, and I told you that microfilaments or protein fibers are going to be formed. So protein fibers are formed from dimers. What are dimer? Dimer are made from tubulin. Two types of tubulin, alpha and beta, joined together to make one dimer. These dimer are merging with one another, like the monomers are grouped together to make a polymer. In the same way, the dimers are joined together to form a long fiber, which is called the protofilament or microfilament, and they are actually spindle fiber. So these spindle fibers are originating from here and grows toward the chromosomes. This is the ultra microscopic structure of the spindle fibers. They just radiate from every side, not necessarily to form toward the chromosomes, but they are present in all the direction. And this initial formation of the spindle fiber makes a shape. This shape is called aster. So aster formation takes place in prophase as well. Similarly, dimers will also be originating from this sides. These are the spindle fibers to be formed, and spindle fibers are formed from the tubulin protein. Tubulin protein is actually making dimer, and they are growing toward the chromosome. So this is the end of prophase actually. So what's going on in the prophase at the end, that we can see that there is no nuclear membrane, there is no nucleolus, the centrosomes are migrated to the opposite pole, and each centrosome have the ability to generate the spindle fibers or microtubules in the form of dimers made up of tubulin protein. But these protein fibers will not reach to the chromosome. They are toward the chromosome. They are growing, slightly growing toward the chromosomes, but will not attach to the chromosome. This is the identification of prophase. Once they join the chromosome, then it means we are in metaphase. So in metaphase, chromosomes are linked to the spindle fibers, but here they are not reached, but their development have been taken place, and they are moving toward the metaphase. So these are some important and major steps of the prophase. We see that the two centromeres are migrated to the opposite pole of the nucleus, and finally we see that the formation of spindle fibers from mTOC. mTOC means centromere. Centrosome. mTOC means microtubule organizing center. Microtubule organizing center. They are the structure which organize the microtubule. This is called centrosome. So formation of spindle fiber from the centrosome, but not complete. It's not complete, does not reach to the nucleus, to the chromosomes. So these are some important steps. And now let's see what is going on here in mitosis and prophase to be discussed here. So prophase, here is the step of prophase, and what does it say? By the start of prophase, the chromosomes are double structure. Because why they are double structure? Because during the S phase, each DNA has been replicated, and the replicated and the older DNA join together to make one chromosome and become condensed. Because the S phase of interphase, DNA has replicated the two chromatids. Chromatid is also a condensed form, but it has only one DNA as its separated one. It is only only the centromere. The two chromatids making up the chromosomes, the two chromatids, sister chromatids joined together to make one chromosome, are held together by centromere. It's a big identification of prophase. The chromatin becomes condensed. Chromatin means the DNA and histone in the uncondensed form, because chromatin always use the word chromatin for uncondensed DNA. The chromatin becomes condensed during prophase. Here the condensation takes place, but keep in mind condensation never complete here, because in metaphase the chromosome becomes further more condensed. Still it is the process of condensation, with the result that the chromosome becomes shorter and thicker. Each of the two centrioles or centrosome, you can say, move to the opposite pole of the nucleus. The nucleolus disappears and the nuclear envelopes begin to fragment it and disappear. Toward the end of prophase, spindle fibers appear, some of which attach to the centromere of the chromosome. This is not actually the case in all the time. There sometimes, in few cases, some of the spindle fiber even reach to the chromosome and become attached, but not all. So we say that the spindle is not completed, although some of the spindle fiber even attached to the chromosome, but very little number. Majority of the chromosomes are not reached. Majority of the spindle fiber are not reached to the chromosome. So toward the end of prophase, now these spindle fibers radiate from a structure at the two poles and called the microtubule organizing center. They are talking about the centrosome like in aster-shaped structures. These organize the tubulin molecule. What are tubulin? I told you, protein molecule, into microtubules, which in turn form the spindle fiber. Microtubules means protofilaments. Many dimers have been held together, just like a chain of your watch have many strips together. The strips are said to be the tubulin, the dimers, and they join together to make the microtubules. That's the end of prophase. And what we will discuss in the metaphase, that's something very important, because two steps in the mitosis are very in detail, and we have to point out some important idea. These are the metaphase, prophase, to some extent always anaphase. But telophase is not so much complicated, because telophase is the reverse of prophase. All the steps of prophase will be reversed in telophase. We will continue this process of metaphase in the next video. Stay with us. See you in the next video.